Okay, so we've been talking about Mendel and the experiments he did with pea plants, and some of the different hypotheses and theories and laws that he created. So one is the law of segregation. The law of segregation, when you followed one character, for example, the flower color, he would be able to see, the first generation, the parent generation were homozygous for that trait. They either had 2 copies of the purple gene or 2 copies of the white flower gene. And then the first generation, the F1 offspring, were monohybrids. So they were hybrid or heterozygous for one character. For that one character, they had a purple gene and a white gene. So from one parent, they inherited a purple gene, and from one parent, they inherited the white flower gene. And when you take those 2 monohybrids and cross them, when you cross 2 heterozygotes, you have a monohybrid cross. And that's what created the F2 generation. The second law, the law of inheritance, was when he followed 2 characters at the same time. So he has this parent generation, he has seeds that are yellow and round. So they have 2 copies of the yellow gene, which is dominant, 2 copies of the round gene, which is dominant. The other parent has seeds that are green and wrinkled. So they have 2 copies of the green gene, which is recessive, and 2 copies of the wrinkle gene, which is recessive. And when he crosses them together in the hybrid generation, the F1 generation, it's hybrid for everything, right? It has a yellow gene, a green gene, a wrinkle gene, and a round gene. And then when you put those together, are you going to end up seeing only yellow that are smooth and green that are wrinkled? Or can these traits independently assort? Can you get them independently? Which means you would see yellow and round, green and round, yellow and wrinkled, green and wrinkled. So do those 2 traits have to always go together? Yellow always means round, green always means wrinkled, or can they go separately? And the results that he actually saw, these are the numbers that he had. He saw this result. So he saw that you don't have to keep those 2 traits together. The chances of being yellow and the chances of being green were 3 to 1, and the chances of being smooth or the chances of being wrinkled were 3 to 1. And so then the chances of that, you get 9 to 3 to 3 to 1, okay? So green and wrinkled was 1 out of the 16, yellow and wrinkled was 3 out of 16, green and round was 3 out of 16, and yellow and round was 9 out of 16. So if you do a hybrid cross, you can see, because these go independently of each other, these are all the combinations you can have. So I'm not going to read through this chart, but you can pause here and look at all the different cases where you get each different phenotype showing up. So this is a dihybrid cross. This is a cross between F1 hybrids or F1 dihybrids. So 2 traits that we're looking at, the color and the texture of the seed. Okay, so using this dihybrid cross, he developed the law of independent assortment, which says that each pair of alleles segregates independently of any other pair of alleles during gamete formation. So the trait for seed color, you have a yellow gene and a green gene, they can go either way. And then the trait for seed texture, if it's smooth or wrinkled, can go either way. That one doesn't control the other. They are independent how they're going to sort out. This only applies to genes on different non-homologous chromosomes or that are far apart on the same chromosome. So if that gene for gene color and if the gene for seed color and the gene for seed texture were right next to each other, they probably would always sort together because they're right there on the same chromosome. But because they're on different chromosomes, and he lucked out that he studied traits that were all on different chromosomes. Because they're on different chromosomes, they can sort oppositely from each other. But genes on the same chromosome that are near each other are likely to be inherited together because there's not really a way that they could separate from each other, right? And so that's why sometimes you'll see some traits like if you have this blood type, you're more or less likely to develop this trait. Or if you have this ethnicity, you are more likely to have this disease. Because some genes are going to be close together. So sometimes having one gene does predispose you to have another gene, but for genes on different chromosomes, they independently mix out independent of each other. So his laws of segregation and independent assortment are based on the rules of probability. So if you are flipping a coin, it can land heads up or tails up. You got a 50-50 chance. So you flip it once and you get heads. If you flip it the second time, you're not more likely to get heads because you got heads the first time. You're not more likely to get tails because you got heads the first time, you still have a 50-50 chance either way, right? So every coin toss is an independent chance of the last one. And that's the same way that the alleles segregate into gametes independently of other genes' alleles. Okay, so let's talk about the multiplication and the addition rules. This might be kind of wordy, so stop and kind of think about it until we're actually kind of working on it. So the multiplication rule says that the probability that two or more independent events will occur together is based on the product of their individual probabilities. So if you're looking at that coin toss, the chances of having heads up one time is 50-50, 1 out of 2. The chances of having heads up the second time is 50-50, 1 out of 2. If you want to know what are your chances that you get heads up both times, you'll say 1/2 times 1/2 is 1/4. So you have 1/4 chance that both heads up or both tosses are going to be heads up. And so how likely you have an F1 monohybrid cross, you can use the multiplication rule. So each gamete has a 50% chance of carrying the dominant allele, and each gamete has a 50% chance of carrying the recessive allele. So that's flipping a coin for each individual gamete and then multiply them together. The addition rule says that the probability of having two or more exclusive events occurring will be calculated by adding together their individual probabilities. So if you have, well, we'll look at some examples in a second because I don't want to confuse you at this point. So you can use the rules of probability to predict the outcome of crosses with multiple characters. So instead of having to draw out a dihybrid cross every time you want to know how likely is it that this person will have type A blood and yellow hair, or how likely is it that this person will, be lactose intolerant and able to taste this chemical. You don't have to draw out a dihybrid cross. You can just use these rules of probability. I do want to say right here, this 1/6 should say 1/16. So if you want to know the probability of having a gene that is YYRR, you're going to say a 1/4 chance that you have two dominant Y alleles, and 1/4 chance that you have two dominant R alleles. 1/4 times 1/4 is 1/16. Okay. The probability of having YYRR is going to be 1/2, and that's based on the addition rule. So you could have the dominant from your mom and the recessive from your dad. That's a 1 in 4 chance. You could have the recessive from your mom, the dominant from your dad. That's a 1 in 4 chance. 1 in 4 plus 1 in 4 is 1/2. So you have a 1/2 chance of having this heterozygous allele, and then a 1 in 4 chance of having that your mother and father both gave you a dominant allele. So 1/2 times 1/8 is 1 in 8. So the probability of having this is 1 in 8. And you can actually go through this with 3 or 4 or 5 traits. What is your likelihood of this? What is your likelihood of this? What is your likelihood of your third trait, your fourth trait, whatever? And you can go through and calculate your probability of having any combination of genes based on 2 parents. Okay, sometimes inheritance patterns can be a little bit more complex than what we've looked at. So the relationship between genotype and phenotype isn't always just a 2 traits, dominant, recessive, 3-to-1 ratio. This is because many heritable characters are not determined by only one gene with 2 alleles. There could be multiple genes and there could be multiple alleles. So like blood type, you could have type A blood, you could have type B blood, you could have type AB blood, you could have type O blood. There's multiple alleles at play in there. The A, the B, and the O allele, right? So different alleles. But the basic principles of segregation and independent assortment will apply even when we talk about more complex patterns of inheritance. When we are looking at single genes, the inheritance of characters by a single gene sometimes can deviate from Mendelian patterns in some situations. One is when the allele is not completely dominant or recessive. So, in the pea plant colors, it was purple or white. It either made the purple gene or it didn't make the purple product. But what if the 2 different alleles are making 2 different proteins and then they're interplaying with each other? So sometimes you don't have completely dominant or completely recessive alleles. Sometimes a gene has more than 2 alleles. So we talked about blood type. And sometimes a gene can produce multiple phenotypes. And then also sometimes we see more than one gene applying towards towards a certain allele. So let's just talk through some of these. Complete dominance is when the phenotype is the heterozygous and the dominant homozygote are identical, right? So purple flowers, whether you have 2 purple genes or 1 purple gene, you are purple. Incomplete dominance means that the F1 generation is in between. So this picture is in between. So a red parent and a white parent, the red and white both work together to contribute to pigment, and you end up with pink. So that is incomplete dominance. Neither one is completely dominant. They both are dominant together, blending towards this. And then in codominance, you have 2 different alleles that affect the phenotype in separate, distinguishable ways. So in codominance, you may see some flowers that were red and some flowers that were white on the same plant. You may see a flower that has some red and some white flowers on it, or that has red and white parts on its petals. So those would all be codominance, when you can see red in distinct ways and you can see white in distinct ways, both in that same plant, that same organism. They are both being expressed in separate ways. So the relationship between dominance and phenotype is going to be kind of based on the biochemistry, what's going on with the actual genes and the actual products. So in the case of pea shape, the dominant allele coded for an enzyme that converted starch to a different form, and the recessive allele didn't convert that form. So you end up with a whole lot of unbranched starch, which takes water in and then wrinkles as it dries. So that's why that's recessive, because you're just accumulating that unbranched. If you have 1 gene or 2 genes to make that functional protein, then you aren't accumulating that unbranched starch. So your seed is going to stay nice and round. Okay, let's talk about another disease, Tay-Sachs disease. Tay-Sachs disease is when you have a dysfunctional enzyme, and that dysfunctional enzyme will result in lipids accumulating in your brain. Lipids are fats, and you don't want those lipids accumulating in your brain. And this is pretty fatal. At the organismal level, the allele is recessive. So if you have a dominant allele, you are going to not get to the point where you are developing disease. At the biochemical level, the phenotype, or how the enzymes are working, is incompletely dominant. You see some of your chromosomes are going to be making the protein and breaking down stopping the production of lipids, and you'll also have some dysfunctional enzymes being expressed. So you're able to see some enzymes functional and some enzymes not functional. It's codominant at that biochemical level. At the molecular level, the alleles are codominant. So you are seeing some alleles, all the alleles are producing independent proteins, independent of each other. When we're talking about recessive and dominant, we are almost always only going to talk about at the organismal level, because that's the level that really makes a difference. So we would say Tay-Sachs disease is a recessive allele. And then also let's note that dominant alleles are not necessarily more common in populations than recessive alleles. One example is this condition called polydactyly. Polydactyly is a dominant allele. Polydactyly causes you to have extra fingers or extra toes, but it is not common. We only see about 1 out of 400 babies in the United States born with polydactyly. So it's not common, even though it's dominant, okay? So please don't confuse dominance and recessive with how common in the whole generation it is, okay? Just because very few people actually have that gene to pass on. It doesn't occur, that doesn't occur in the general population frequently enough to make it common. So being dominant doesn't mean that you're common, even though if you cross 2 heterozygotes, the dominant allele is going to be common, okay? So in the real world, we are not always crossing heterozygotes to have dominance does not correlate to common frequency. Okay, and then multiple alleles. Most genes exist in populations with more than 2 alleles in it. You have more than 2 versions of it. So we've talked about the ABO blood group. We have 3 different alleles. One is the allele for A type. So that if you have the A allele, we're going to write it with a capital I and then a subscript A. That subscript A is capital. So because the I is capital, It tells you that this is, these are going to be codominant with each other. And then capital A, because you're producing the A enzyme, which means that you're going to have this sugar stuck all over your blood cells. If you have the B allele, you write it as a capital I with a subscript B. And that means you're going to make the B carbohydrate and stick that on your blood cells. If you don't have either one of those, some alleles don't make any sugar. That is a lowercase I because that is recessive. So if you have an allele to make the A-type blood, you are going to have that A protein, that A sugar on your genes. If you have the allele to make the B sugar, you have the B sugar on your proteins. You have the B sugar on your red blood cells. If you have inherited both, you have an allele for A and an allele for B, you are going to express both of them, okay? And then if you have two recessive, you are not going to express either of them. So if you have A-type blood, you are making only the A protein. If you have A-type blood, you're making only the A sugar. If you have B-type blood, you're making only the B sugar. If you have AB-type blood, you're making the A sugar and the B sugar. And if you have O-type blood, you are making neither sugar. Okay, so that's having multiple alleles, different types that both can be dominant or recessive depending on the situation. And then many genes have multiple phenotypic effects, and this is called pleiotropy. So having one gene means you're going to have all of these effects happening. So many hereditary diseases such as cystic fibrosis. Cystic fibrosis is caused by one gene, but you have a lot of side effects of it. Sickle cell disease is one gene, but it gives you a lot of effects. You can see a lot of different things in it. And some traits are determined by two or more genes. So let's talk about epistasis and polygenic inheritance. These are both going to be situations where more than one gene determines one trait. In epistasis, one gene is going to control another gene. The gene product of the protein produced by one gene controls how the second protein product acts. And in polygenic inheritance, multiple genes work together independently to affect a single trait. So let's go through both of these. So in epistasis, the expression of one gene at one locus affects the phenotypic expression of a second gene at a second locus. So in Labrador retrievers, they have, and a lot of other mammals too, but we're going to talk about Labrador retrievers. Their coat color is dependent on two genes. The first gene is going to control what pigment color is made. So it might make black or it might make brown. So we have a black allele and a brown allele. So you can have black or brown Labrador retrievers. But there's a second allele. The second gene, which either produces a dominant or recessive, determines if the pigment is deposited in the hair. So this first gene is controlled by the second gene. You could make lots of black pigment, but if that pigment doesn't get transported into the hair, it's not going to show up. So this E allele is going to control if they have any color at all. So you see when you have this two copies of the recessive E trait, these Labradors are always going to be this kind of yellow color. You're going to have, I just forgot what that color is called, I'm sorry. But they're not going to be black or brown. They're going to just have this kind of uncolored fur color. Whether they are making the black protein or they are making the brown protein, they are not going to have any color. But when they have at least one copy of the functional E gene, they are able to transport that color in, and then you can see them developing black or brown color. So that is epistasis. This E gene controls the B gene. So being able to control where the pigment goes in and depositing that pigment in their hair and not just making the pigment. Here this goes through the genetic background of that. Yellow labs, that's what I was trying to think of. Black, chocolate, and yellow labs. So pause and read this. I'm not going to read it to you, but pause and read this so you can understand kind of how we get that 9:3:4 ratio instead of a 9:3 to 3:1. So a 9:3 to 3:1 is what you would expect in a perfect dihybrid kind of ratio, right? Instead, you get this 9:3:4 because it is epistatic. Epistatic interactions produce a variety of ratios, but they're all going to be modified from this 9:3:1. So you don't need to memorize this ratio because that changes depending on what the epistatic interaction is. Okay, then let's talk about polygenic inheritance. This is when one trait is controlled by many genes. So quantitative characters are those that vary in a population along a continuum. So height, people aren't all tall or all short. There's a whole variety of people that are fully grown. Skin color, we aren't all just one or the other. We are in a range of skin colors. And that is because when we see ranges, we are typically going to be talking about polygenic inheritance. So it's not just the gene is this or that, but it's this gene is this or that, plus this gene is this or that, plus this gene, plus this gene. And when you add up all of those, you have a whole broad range. So for height, you have over 180 genes that affect height. For skin color, here they're looking at 3 different genes. In reality, there's a lot more than 3. But you can see how if you have like the recessive form of all of them, or the unpigmented form of all of them, all the way up to the pigmented form of all of them, you have a range from 1 pigment to 2 to 3 to 4 to 5 to 6 that can affect your gene color, that can affect your skin color. Okay. And then let's not forget nature and nurture. So the environment you are in can affect your phenotype. One obvious way that nature and nurture is if I go and dye my hair blonde, or if I go and dye my hair red, my hair color is no longer being determined by my genes, but it's being determined by my environment. If I am going out in the sun, I can get a tan. That is not my skin color depending on my genes. That is skin color depending on my environment. Okay, so many traits can be impacted by your environment and not just your genes. And the phenotypic range is going to be broadest for polygenic characters. And when you have traits that depend on multiple genes and environmental influences, it's multifactorial. So multi means a lot, many factors. So many different environments, many different genes can all affect how that trait is looking at. Another example here is hydrangeas. So if you grow your hydrangeas in basic soil, their flowers are more likely to turn pink. And if you grow them in acidic soil with free aluminum, they're more likely to have blue flowers produced. So that's not just their genes. You could take this plant and acidify their soil, or take these plants and make their soil more alkaline, and you would see the colors change, independent of their genes. So an organism's phenotype includes its physical appearance, its internal anatomy, its physiology, and its behavior. And its phenotype reflects its unique genotype and its environmental history. So how tall I am depends not only on my genes that I have, but also if I was exposed to proper nutrition throughout my childhood. Okay, we're going to pause here to start the second part of this lecture video.